for the invite, and thanks everyone for being there. I have to give you two warnings. First is that my internet is sometimes a little bit unstable, so I hope that will go well during the entire hour. And the second is that I'm a bit allergic, so I might have a burst into sneezing at some point, but I hope that doesn't happen either. So as Benjamin has pointed out, I come from kind of machine learning and statistics background. Before that, I did a computer engineer degree. So I only moved to neuroscience after my PhD, where I've been developing methods and ways to look at the data in neuroscience. So during that time where I've been working on these kind of things, I've been slowly learning a few things about this type of work that I wanted to start with, to share with you. It's just common sense, but I thought it will make sense to mention this. And the first thing is that when one does methods developing for neuroscience, it is very important to keep in mind that we don't do methods for the sake of doing methods. So methods should be at the service of the neuroscientific question, and that's something that not everyone that works on methods always remembers. So I think it's a good thing to bring forward. And for that reason, another thing that comes from that is that your methods should be as complex as necessary, but not more. So you can think of complexity and complicatedness as a sort of a budget that you have to wisely administer and spend. So those are two things that are important, I think, when we work with methods development. And I will also make another point, which is something that is not often taken into account, but while it's true that the important thing is the neuroscience behind, and that's what we work for, that doesn't mean that methods, the choice of methods, is not important. I think that when we decide to use certain types of methods instead of others, we're also maybe unknowingly, but we are also making a point about our philosophical understanding of how the brain works. So there is almost a belief implicit on the methods we choose, and that's an important thing from an application point of view. So I wanna keep these two things in mind during the rest of my talk. So in particular, when I've been developing methods for these things, there's two things that I wouldn't call them beliefs, but a kind of a way of thinking about the brain that are important and are at the essence of these methods. And one is that, well, that doesn't seem very controversial, but the brain is a very distributed system. So cognition, for example, can only make sense if you consider the brain as a very interrelated system. The second one is that it also seems like obvious and common sense, but you will see that not always, sometimes in practice, we are a little bit at odds with these things in practice when we do standard analysis. And the second one is that brain responses are meaningfully variable, and you will see what I mean by that. So that the brain is a very distributed system, very few neuroscientists nowadays will disagree with. So there's a lot of studies that show that the communication between regions is critical, above and beyond of the activity within a given region. So this integration across regions is critical for many aspects of cognition. So this is just one of many studies that I only put because it's ours, where we showed that in order to when you want to predict a lot of psychological traits and clinical mental things that relate to mental health and stuff like that, often it is the actual interaction between regions that predicts best, above and beyond the activity on these regions separately. So we published this in this paper. But even though that is, I would say, like common sense in the neuroscience community, the dominant approach to look at brain data is still like like this one in this slide. So here, it's just a common experiment where you have, like, for example, two types of stimulus, faces and shapes, and then you model how the activity in each region separately differs between these two conditions. So you do that for each region separately. In a way, you are not really embracing this view of the brain as a distributed system. So I would like to develop, so one of my aims is to develop methods and models that are a little bit.
Excuse me, Diego. So since we are not so familiar and we are not so numerous, can you give more details about the experiments? Or
this is just an example, but this is a neuroimaging, like fMRI experiment where you have two conditions. So people are looking at faces or shapes, or you have people that are doing something else, and then you do what is called a GLM, which is general linear model. In neuroimaging, that's not the same than generalized linear model. But anyway, the point is that you do some T statistic for each voxel according to how that voxel differs between the two conditions, and then you draw a map of brain activity of the difference between one thing and the other. So that's like the most common approach for analyzing neuroimaging data, fMRI or other things, is this. But so that ignores by definition this distributedness of the brain. So you look at every voxel, but you don't look at interactions between voxels. You don't look at how these things come together to distinguish between one condition and the other, for example. So that is just one example that even though we all know that the brain is a very distributed thing, our methods sometimes do not acknowledge that. But just that's an example. The other thing is that the brain responses are meaningfully variable. So if you present a stimulus, like a visual stimulus, very simple visual stimulus, and you measure the response of the brain, is different every time. So that variability, we know, again, it's not very controversial. You ask neuroscientists about this, they will all acknowledge that this variability reflects a purpose, reflects a history and a context. For example, this paper here shows that, for example, in visual cortex, you can very well predict a lot of variability on the responses of individual neurons based on the behavioral state of the animal. Things like what the animal is doing, or what is the size of the pupil, or movement, even though it's visual system and it's not related to what the animal is watching, what you can still explain a lot of variance on these neurons from many other things that are not vision. So that is just it just proves that there is a lot of variability that depends on many things and not just the stimulus. And this variability is meaningful. And that study is talking about other things that relate to the variability, but there are many other things that we cannot measure, but that doesn't mean they're meaningless. So we know that variability is something that means that is meaningful in a biological sense, but most of the studies that look at the stimulus processing, what they do is they just look at the average. So for example, this experiment is just some electrophysiological data, EEG probably, where we have is probably records on one single electrode, and you have again, two conditions, faces and cars, and there's a response across trials for each of those two stimuli. And then what people do is just to get the average of faces, the average of cars, and say, okay, that's what it is. But every variability around is regarded as noise. But that kind of insults this understanding that variability is something of the essence. So we evolved as animals to be variable. That's part of what we are. We cannot ignore that variability. So that's the two things that are important to me when I develop methods. And of course, all of this only makes sense in the light of behavior. So the methods I'm going to talk about. The ultimate purpose is to try to relate these patterns that these methods pull out to some sort of behavior. So that can be two different things. Can be behavioral traits, as I mentioned before, like I don't know, IQ, for example, or how healthy you are mentally in different dimensions. And another thing is instantaneous behavior. So which direction the dog is going to go at every given moment. So these are two things, two dimensions of behavior that we want to take into account. So we need methods that are able to model the dynamics of how regions synchronize and communicate. So we need to model network activity, and we need to acknowledge the variability of these brain responses, even for the simplest stimulus. Even for the simplest stimulus, the brain is never going to react the same. And we need to take that into account and model it and extract the most of it. And of course, we need to relate these brain patterns to behavior in some way, in some meaningful way, because that's the point of neuroscience. May that be in actions, stimuli, decisions, or population variability in some particular dimension. And normally, in order to get a better understanding of brain function, we need to integrate across different scales and modalities. So the methods that I'm presenting are designed to work across different types of data. So the way of interrogating network activity or communication in the brain, what we call functional connectivity, the most common way, the simplest way, is to use what is called a sliding window. So you imagine that this is MEG data or EEG data or fMRI data, some sort of temporal signal going on up and down. So you choose the size of a window, and you measure correlations between areas, and then you move the window again, and then you get sort of a moving picture of how regions communicate with each other in whatever metric you're choosing. So that's fine because it's simple, and simple is good, but how there's some issues, some important issues. So how do you choose the width of the window? Because if you choose it too short, the estimation is going to be unstable, and if you choose it too long, you're going to miss quick changes, which maybe is what you're interested in. And as a consequence of this window size, time resolution is limited. So you might, whether or not the size of the window you choose, you might miss the faster changes. And of course, it makes an inefficient use of data because you just look at one window at a time. So you don't look at the whole data set, you just look at one thing. So can we do better? I think we can. And the approach that I've been working on as a general framework to get around these issues and others is based on brain states. So instead of going one window, a sliding window like one time after another, we assume that we can describe the brain data as a discrete number of states that go on and off, change from one another, such that each state is characterized by a certain pattern of brain activity. So the way the model, the general framework that I've been working on for years is the hidden Markov model. This is a model that I haven't invented. So it's something that has been around since the 80s. So people have used it a lot in engineering applications, in speech processing, also in genetics. So it's a widespread model in the machine learning community and probabilistic science community. And I've been trying to adapt this to the specifics of neuroscience. I'm going to tell you a little bit more about this now. So the hidden Markov model has three basic elements. And the key thing is that, well, this is a Bayesian inference, it's a probabilistic model. And all of these three things get inferred from data simultaneously. So this is some estimation process that come up with all these parameters without us having to decide anything other than a few hyperparameters. So one thing is when the state activates. So for each time point, you have a probability per state telling you how probable is that state to be active at each time point. So that's when the state occurs. So what are the states themselves? So what are the spatial properties of the states? So each state is a probability distribution that can be many things depending on the type of data you are trying to model, and that can encode information about patterns of activity or functional connectivity or spectral properties and so on and so forth. And then the last element is, well, because the HMM is a dynamical system, so it's a very simple dynamical system, you have these dynamics encoded as a transition probability matrix that tells you the probability of going from one step to another at each time point, or to stay in the state that you are currently. So that's the very basic hidden Markov model, and there are a lot of varieties that sort of, build on that, but that's like the basics. So the benefits of that is that we do not need to specify the window length, because there is no window. We can access the fastest timescales because the visits to the states can be as short as the data prescribes. And it makes an efficient use of data because you train the model on the whole data. So that means that while if you have a difference between two sliding windows in terms of network activity, you can never be sure whether these differences are not due to a noise in the estimation, a statistical noise. In this case, differences between two HMM states are very unlikely due to a statistical noise because you have a lot of data. So they are always differences that come from data. So for example, it's a HCP data, it's a large data set that has a lot of subjects and a lot of time per subject. So if you train an HMM with 12 states, you will have around 70 hours on average of data per state. So that's an incredible amount of data, whereas a typical sliding window is 1 million. So that gives you an idea of how powerful in terms of being precise in the estimations this model is on models that are like this.
Can you say something about how you
Yes.
identify the different states?
That's the task of the inference. So I didn't want to go into the inference, but so there are many approaches to infer the parameters of this model. That means both when the states become active and the states themselves. There's a lot of approaches for this. The one I prefer and use the most is called variational inference, variational Bayesian inference, and basically is just the whole idea of you start sort of randomly and then you estimate. So given a random assignment of state activations, you estimate the state distributions, and then you given these two things, you estimate the state transition probabilities, and then you cycle between the three elements until you converge into a solution. So you fix two, you estimate the other, then go to the next one, so on and so forth. That is called variational inference. It comes from the earlier work of Euler as a mathematician. So that's like a very old thing. But there are other ways based on sampling and other techniques. So there's a lot of ways of doing this. That's a lot of the field in itself, how to do the inference of this model. So I didn't want to do too much into this, but that's the basic idea.
And sorry, and how many states do you end up with for a typical experiment?
That's your choice. So like the basic model, you choose the number of states. The more states, the more detailed view you have in the data. The fewer states, the more reliable the estimation is. But you obviously you capture less information, obviously. And there are models like extensions of HMM that can infer automatically the number of states. They call it infinite HMM. I wouldn't recommend them in neuroscience because they are very hungry in terms of time and data, and those things for neuroscience are not always practical. So I think the best approach is to run HMM with different number of states and compare your conclusions that you get for these different runs across different choices of the number of states. I think that's the most sensible to me.
And is it usually like 10 or 100, say?
But it really depends. So it's like you know ICA, right? So how many components do you run in ICA? So that's depends on the application. So like you are talking about fMRI preprocessing or fMRI, like pulling out networks in fMRI data, I would say 50 ICA components. Maybe with HMM would be 20 HMM states. That would be reasonable for a big fMRI data set. But if we're talking about animal data, we'll have terabytes of data with just a few regions. Maybe you can get many, many more. But that it really depends on the data. And on how you parameterize the states, obviously. So you can have more complex state distributions and you need fewer states. So that's a bit of an art in a way to choose these things. Like many other things like that, actually. So, so talking about this, so most of my work in these years have been to develop into developing different flavors of this approach for different types of data, and that every type of data will have different answers to that question. In particular, I worked with human data mostly, but also a little bit of invasive electrophysiology in animals. Right. So before going into example, so I so just to give you a taste of the kind of things that how am I out of time? The kind of applications that we found this XMM useful for, I'm going to give you 4 quick examples. So it's not my intention to go deep into the neuroscience behind these things. It's just like a bit of a taste of what the things that you could pull out with this model. So I will go relatively quickly through those. So the first one is about, so there's a lot of work on understanding the different brain networks when people are at rest. So we're not doing anything. So there are a lot of correlations in between regions in the brain. And there is that defines a few brain networks that are very well studied, like, for example, the deformal network or the attentional network or the visual, the auditory, and so on and so forth. And there has been a lot of research in the fMRI community in studying the spatial properties of these networks. So how these networks differ between them, how these networks change in a clinical population, and so on and so forth. There is not that much research or understanding on the temporal properties of these networks. So these networks obviously, fluctuate also in time. So they activate and deactivate, and these dynamics, these temporal dynamics, are not very well understood when people are at rest. So we are doing a task. The task kind of defines or constrains this temporal dynamics. But when we're at rest, how this network activity unfolds is not very well understood. So we investigated that question using the HMM on a big data set, which is the Human Connected Project. So it's around 1,200 subjects, 1 hour data each. As I said, this is fMRI. So we ran this HMM model with 12 states in that case. Well, and also different numbers, but I'm going to focus here on 12 states. And one of the things that were more remarkable about this decomposition into networks using the HMM, where, as I say, every network is defined as a pattern of activity and also as functional connectivity, so coupling between regions, is that transitions between networks were really not random, which you would expect because the brain is not fully random. But not just that, but the pattern of transitions between networks was actually quite not rigid, but quite constrained in a way that naturally the composition splits into 2 groups of states, such that transitions within a group are much more likely than transitions between groups. So we call these groups that came in a completely data-driven way, came completely from the data. We call them metastates. So groups of states, groups of networks. So there is quite a strong property on the temporal characteristics of these networks that we pulled out with this model. We can look at the special properties of these groups of networks, and we found that one of these metastates is characterized by regions that are typically characterized by higher cognition, regions related to so to say high cognition, like, for example, the deformal network, which is supposedly related to introspection and kind of reflection, or language networks, or attentional networks, whereas the other metastate, the networks that belong to the other metastate, were more related to more sensory motor functions. So we found this separation at the temporal level. So we already knew about this separation at the spatial level, but we know now that you tend to switch between groups of networks that are more cognitive, and every now and then you change to something that is more sensory motor. And that's without making a mechanistic hypothesis of why that is. That was something that came out from data. So we found that.
Sorry. I just, before you go on, can you go back just your previous slide? It's very to grab the main idea to. So you took the data which was already in the Human Connectome project, and within this data, you were able to, with your statistic methods, differentiate between groups which activate in resting state or introspection. That's it, right? That's the global picture.
So it's a group level. So you infer the model at the group level. So you have the states that are shared by all subjects, but every subject has their own state activations. So every subject have their own train of thought, obviously. So every subject have their own temporal dynamics, but they all share the same states. You could also do this at the individual level. It's a bit more complicated, but you could do it too. So the point here is that the networks of the states, the way they switched across them had a specific logic to it. And that was a group level effect. That happens for all subjects. But I'm going now for more about the group level thing. So the time every subject spent on each of these groups of networks, so metastates, had a significant relationship with a lot of psychological measures. So that gives you at least that this is not just an artifact made by the model, but means something, given that these behavioral traits were not seen by the model in any moment. Furthermore, these metastates are very subject specific. So because subjects were scanned several times, so if you tend to spend an amount of time into one metastate, one scan, you will do the same in the next scans. So very subject specific trait or signature. And also this metastate profile, how much time you spend in each, is very heritable. So people in that data set, there were entire families being scanned. So you have brothers and sisters and twins and stuff. So if your twin tend to spend that much time in one of these group of networks, you probably do similarly. Okay, so that was one example. So I'm going to move on now.
Can I ask for clarification? Okay. Yeah. When you talk about a network, are your nodes representing the regions of the brain, or are they representing brain states? Which one? What is?
Yeah, good question. So we run the HMM on ICA components. So its state is defined over a set of brain regions that we pulled out using ICA, so that data-driven parcellation. So its state is defined by some interaction between these regions. So this graph here relates to transitions between states. So each node here is a state, and each state is defined as a pattern of interaction across regions. Sorry, I wasn't very clear on that. I realized I wasn't very clear on that now. Thank you. Is that clear now? Thank you. Right, so that's the resting state in wakefulness. We are interested in the other resting state, which is sleep. So as you all know, the sleep is divided into 4 to 5 sleep stages. So the sleep stages have been defined or known since many years ago, like probably 70 years ago. And they're typically, typically the way that this is sleep research works is that someone who knows about sleep looks at EEG data and other things in windows of 30 seconds, and then look at the properties of these 30-second windows and label this window as one of 5 sleep stages. Could be REM or shallow sleep or deep sleep and so on and so forth. So we're wondering whether we could do something more data-driven, so we could have more data-driven description of sleep. So we use fMRI, and we did exactly the same thing that we did in the previous piece of work while people get sleep. So they go through most of the sleep stages. They don't go to REM sleep, but they go through the others. And what we found is that we pulled out networks of activity that were very predictive of the sleep states as defined by an expert, but they were more informative in a way because it was a more fine-grained description of sleep. So we found models of networks that you transition within more often than across. And these models of networks, same that in the other work, these models of network corresponded to the original sleep stages, but we now know which are the dynamics within each of these modules. And that was an additional layer of information on top of this like well-known traditional view of sleep. So we were able to infer a few things about the nature of sleep using this model. So that was another example. Again, I don't want to go into a lot of details because actually I've covered like 1/10 of my talk, and I'm only half an hour. Oh, no, actually 20 minutes. So that was one thing. We are very interested in MEG in particular, so magnetoencephalography, because fMRI is fine, but we have an interest in as you have noticed, we have an interest in the temporal characteristics of brain dynamics. And fMRI, first of all, it's not a very direct measure of brain activity, and second, it's very slow and is not very easy to make statements about temporal characteristics on fMRI. So we have an interest in working on MEG. So we were interested in applying this kind of model to whole brain MEG activity. So we developed an HMM model that where states were defined as a pattern of power across regions and also phase coupling. So we have like regions oscillate. Sometimes they oscillate in phase. So states were characterized by patterns of this phase coupling across regions and also across frequencies. So we use this to get a bit more of insight into a very well-researched network in fMRI, which is the default mode network. And we found a description of this network in MEG. It's actually in MEG, this network splits into two networks, maybe because you can afford this more temporal resolution, and it has different frequency components and so on and so forth. I'm going to leave it like this because I need to rush. So there was no behavior in this paper. So we were because there's a lot of interest in this network, but what is the behavioral relevance? So there's no behavior. So we wanted to in the next piece of work, we wanted to get a bit more of insight on what is this network that happens when you are not doing anything at rest. What is that for really? So we went into something that is called replay. So it's a phenomenon that is relevant for people that study memory. So when you memorize something, in particular when you memorize a sequence or you memorize how to go from one place to another. So the hippocampus is very important in the process. And then when you stop, so after you learned, there is a phenomenon called replay, and that is so those sequences you learned spontaneously and subconsciously replay, and that's thought to be very important for memory consolidation. That happens both in sleep and wakefulness. So we hypothesized that replay might have a relationship with this default mode network that no one really knows what it's about. So we set to study this with HMM. So we have an experiment where people have to memorize some sequence of pictures, and then we scan them at rest afterwards. So we ran the same analysis that in the previous paper. So we ran the HMM. We found these networks that I've been discussing, these DMN networks. But we also applied some methods that I will not describe to find when replay occurs in the brain spontaneously. And the hypothesis was that these two things will tend to happen simultaneously, and that was the case. So we found that the DMN was sort of a facilitator of this replay. So that was something that requires being quite sharp in the temporal domain. So it requires methods that can really delimit when things are happening. So that was something that HM was very useful at. So these four examples were examples of applications where we used this HMM in an unsupervised way. So we ran on data and we found patterns from data, and we then later on associated these patterns to something that we were interested in, may that be replay or sleep or something else. But moving forward, what I'm actually interested now these days is to how to model behavior explicitly. So this model, this work that is shown so far, we run these models on brain data only, and then we relate this decomposition to whatever behavior post hoc. But there are reasons for us to want to include behavior, to model behavior explicitly within the model. And that's because, you know, a lot of stuff happens in the brain. So there's a lot of things going on. And if you're interested in something that is relatively subtle, like for example, if you're interested in studying imagery is a subtle effect, you want to include the behavior, you want to include the effect into the model, because otherwise you're going to miss it. So the model, the model that I'm going to talk about now has to do with including behavior into the model. So the way people model stimulus processing, for example, or decision making, so the way people include the behavior, that models the relationship between brain data and the behavior, is typically through decoding analysis. So basically, decoding is just the use of some machine learning or statistical approach to predict, for example, in stimulus processing, what the subject is perceiving using the brain data as a predictor. So you take a machine learning model, you feed the data, MEC data, for example, and then you try to predict what the subject is looking at. So you do that. So the way you do this, so you obviously have a lot of trials. So you have this person looking at things for 1,000 times. Then you stack all the trials, and then you go time point by time point. You train a classifier so that when to see that classifier can predict what the subject is looking at. Then you move the time point or the window, and you do that again, and then you sort of have a moving picture of stimulus processing, in terms of both the spatial information when this stimulus processing, where this stimulus processing in the brain occurs and when. So that's fine, but if you remember how we started this talk, I'm going to skip this for now because I'm short of time. If you remember how I started this talk, one of the things is that brain responses are variable. So we don't respond every time the same way. And this method assumes that we do, or at the very least, only captures effects that are common of all trials. So that ignores the variability in the data. So this is just an example. No, I'm not going to go through this. So this is a synthetic data where I just show, without going into details, I just show that these methods can give you a mistaken view of the data if the underlying effects were not time locked. If there is temporal variability in when things happen, this method will give you a misleading view of the data. So these two slides are showing the simulations that I did to show that, but I don't have time for that. So the point is, we should take this into account, especially when you have tasks where you would assume that there's a lot of temporal variability. For example, if you do have decision-making task, you can hardly assume that you make the decision always at the same time. Or if you ask someone to picture something in their minds, that imagery process, you can hardly assume to occur always at the same time. But even when it's something simple, when you are shown something, and the visual cascade is relatively well-defined and stereotyped, even though we know these days, we know now, that this cascade of activity depends, for example, on the state of the brain when you are presented at a stimulus. So we have a really strong interest in capturing this temporal variability, and this method I'm going to present now is about that. So what's the deal? So as you remember, the basic decoding method works by training one decoder per time point across trials. So one decoding model per time point. So the idea here is to have a much fewer numbers of decoders or machine learning classifiers that predict stimulus from data, but we allow them to occur at different time points per trial. So now when these decoders become active is data-driven. So we have fewer decoders, and in exchange, we allow them to come on and off at different time points. So we are trading spatial parameters for temporal parameters. That's the trade-off. Is that clear? Because that's the key point of this. I'm going to present an example to see that that's more clear. I'm not going to finish here, so I'm going to skip a bit of my talk. So we had an experiment where it's a very simple task. So the subjects had to remember certain orientation, and then they were shown orientations. And when that orientation matched the one that they had in mind, they had to press a button. So it's a very simple perceptual memory task. So we ran this model in where we predicted the thing that is being shown, and we found is something like this. So in this case, in this plot on the left, we have trials by time, and every color is a different model, it's a different decoder. So you see there is a progression of models from model 1 to model 5, but you also see that there's a consistent, there's a lot of temporal variability in when you go from model 1 to model 2 and to model 3 and to model 4 and to model 5. We did not impose this sequence of states. It came naturally from data, although you can impose the sequence if that's useful. But the key point is that, for example, at 250 milliseconds after the stimulus presentation, you have most often model 2, as depicted here, active, but not always. So that transition between the stages of visual processing is not always the same. So that was the point of this. And you might say, right, but this is an estimation too. So you are estimating when each model becomes active, and that's subject to error because a statistical estimation, and every time you estimate something from data, you are subject to statistical error. So how do we know that this variability in when you go from one state to another is not just a statistical error? And that would be a very good question. That's why we related these temporal properties of when you go to one state to another to something like reaction time. So we know when the subjects press the button. So we know when they process the information quick, and we know when they process the information slow. So they have to remember if that thing that they see is the thing that they had in mind in the first place, and press a button if that's the case. So there's a bit of a decision there. So we know when the decision is made quick, and we know when the decision is made slow. And there's a very strong relationship between how quick you transit from one state to another and reaction time. So that means that even though there might be, and there is, a statistical noise in this estimation of the temporal properties of this processing, some of this variability is legit because it relates to something that is biological and that the model didn't see, which is reaction time. Okay, so there is the next part of the talk was about how can we relate these models to behavioral things at the population level. So how can we relate this thing to health variables, so to IQ or to to population variability? But that will take me at least half an hour at the rhythm I'm going. So I'm going to skip this. And if anyone is interested in this, we can talk about this some other time. So I'm going to jump directly to conclusions, because I think that's good, because I can leave some time for questions. So overview, I presented an unsupervised HMM with a Bayesian adaptable model to characterize brain patterns that you run on the data. And that data can be fMRI, can be MEG, can be EEG, can be LFP, can be spike in activity, can be many things. And I presented a more supervised HMM where you take into account a specific relationship between brain activity and the stimulus. Please note that these models are not biophysical models. So I know that a lot of the talks that have been going on in this symposium have been presenting biophysical models. These are not biophysical models. These are agnostic models. They are data-driven models. So if you sample data from these models, you will not get data that looks like real data. So that's something you should know. But it's still useful to pull out patterns that you might be interested in one way or another. So my future work, if you ask me, is to try to bring together these two views of the world. So this biophysical model with this more data-driven ways of looking at data, and that's what I'm interested to do in the future. The last point of this overview is some machine learning methods to link these models to behavior, but I didn't talk about that, so let's not dwell on it. And that's it. So many thanks to you for listening.